very good evening, everybody, and welcome to another episode of Eclectica, the different news. Today, I have here as my guest a very visionary person. If you are from the field of pharmacology, pharmaceutical drug development, or biotech, I'm sure you know Dr. Subrahmanyam Vangala, very popularly called as Manny. Thank you, Manny, for accepting my invitation, and it's an honor and pleasure to have you on my podcast. Thank you so much for being here today.
Yeah, it's an honor. Thank you very much. I really appreciate for you to take your time to interview me for this podcast. I'll definitely I'm excited about this and try to see how we can uh, help you understand, not only you, all the audience, what we're trying to do, innovations in terms of the drug discovery, drug development that might be revolutionize the or modernize the drug discovery process in the entire world.
Right. To all my audience, if you are meeting Mani for the first time, he is the founder of Reagene Biosciences and co-founder of Reagene Innovations Pvt Ltd. Let me start with my favorite question, Mani. Please share your journey because I know it's an extensive journey and your vision behind focusing or founding Reagene as such. So that would be very interesting to know.
Right. Fortunately, I'm a biochemist. Before I entered the pharmaceutical industry, as a biochemist, I joined for PhD in Canada, in Newfoundland, with a visionary professor called Peter J. O'Brien, where I actually got to learn about the environmental toxicology, biochemical and molecular toxicology. We used to look at a lot of environmental pollutants and how they cause cancer or how they cause toxicities in humans. And in those days, people used to use animal models extensively to understand the mechanisms of cancer. But uh, that's how my journey started. And then I did my postdoc at University of Colorado Boulder, again into environmental toxicology. Of course, this is all molecular and biochemical toxicology, where we actually look at the mechanisms. It's also called investigative toxicology. The reason for that is that you cannot really dissect a human to understand what is happening in the human. Because if a chemical, you're exposed to a chemical through the lungs or through the mouth or through the skin, you don't know where it goes, number one. And unlike animals, in animals, what we do is we actually can sacrifice animals and look at the pathology, biochemical mechanisms, and everything that you can do. That is the reason a lot of people looked at animals first before going into the humans, in any case, because of the. But I've been continuing that journey also at Berkeley for some time. We looked at free radical mechanisms of toxicology and biochemical mechanisms, basically looking at how, for example, benzene is a major environmental pollutant. How does it cause cancer in humans? Be mind, it doesn't cause cancer, leukemias in animals, even rodents. You take them, dose them, but there's something called a plastic anemia where all the bone marrow cells go down. That's considered pre-leukemia, but it's really, you cannot really dose forever to get leukemia in animals. So very interesting to observation that we made a long time ago. In any case, we had to use animals because we have no other choice in trying to understand the mechanisms, etc. Uh, at the same time, I was exposed to physiological mechanisms of modeling. How do you model the what happens in the humans or even animals? How do you translate to the humans and everything? So my journey ended there in terms of the environmental toxicology, and I switched gears into the pharmaceutical industry. I joined an American cyanamide company, which doesn't exist. It's now part of Pfizer. It was acquired by Wyeth first, and Wyeth was acquired by Pfizer later. So the American cyanamide company gave me a different perspective to look at how do you see the drug behaving in animals to humans, and how do you decide on what is a clinical dose, first in human clinical dose. And these were very interesting phases because I come from a molecular toxicology background, switching into a different thing. And now how you look at is where the drug is going, basically, in the body. And we have done a lot of these ADME studies, we call it, absorption, distribution, metabolism, and excretion studies. These are very, very critical to understand how the drug is going, where it is going, what is doing, is it becoming for producing toxic metabolites, or is it actually going to the target where it can actually work very well. These are the things that we used to look at. And I've been probably a lot of INDs, in investigational new drug submissions over the years. The one question that comes up when you go and try to select a human dose, we follow FDA guidances primarily. I mean, there are also ICH guidances the same similar way, but those are guidances that you have to use a rodent, a small animal, and a canine or a monkey as a large animal. If a drug is safe in both species, then FDA or other regulatory agencies give you a green signal to go into the first in human clinical dose. But when you go to the first in human clinical dose, you always use a conservative dose to make sure that no adverse events or other fatal incidences happen in a first in human clinical trial. In the first in human clinical trial, we only look for a pharmacokinetic behavior of the drug and see, is it actually going into the body and is it actually has enough concentrations to circulate around to go to the target tissues. That is the primary thing in the phase 1 trials people do. But even before you go into that, we see there's a lot of differences. If you take rat study versus a dog study versus monkey study, you see a lot of differences. No animal is foolproof and say, hey, they all behave like humans. So if you get in rat, for example, bone marrow toxicity. In dog, you don't get bone marrow toxicity, you probably get some other toxicity, liver toxicity or something. So then the question comes is the human going to get bone marrow cancer or bone marrow toxicity or liver toxicity? We cannot jump to conclusion. So, but yet, since we are going with a conservative dose and the dose margins that we use in animals versus the humans significantly have a large margin in terms of the therapeutic margin, we try to go very slowly and with an approach that we do. If phase 1 trials are successful, we go into phase 2 trials and then phase 3 trials. But phase 1 trials are mostly safety studies. Basically, you want to make sure the drug is safe in humans. But as I was doing it, but out of 25, 30 compounds, we do the investigational new drugs, maybe 3 or 4 usually go to the 4 first in human clinical trials, phase 1 clinical trials. The rest of them don't make it. They're either toxic, they have other problems, sometimes even chemical issues. We cannot manufacture it, you cannot formulate it. There's several issues which we drop them and take these 3 to 4 molecules into the clinic. The fun starts as you go into the clinical trials. Many times we cannot really, whatever we estimated, the human dose or human pharmacokinetics, is not right on the margin. So in the 1990s, there was a lot of interest in the liver as a drug metabolism thing. In human drug metabolism versus animal drug metabolism, we did a in vitro comparison. Not only me, but many other academics and industry professionals compared it. And we realized that the drug metabolism in animals and humans are different. Typically, humans have only some enzymes, not all the enzymes, which have rats and dogs, etc. So, which brings to the point is that why do we use animals? Because you always are estimating from animals, but you get into trouble in the humans. So, by the way, 90% of the clinical trials fail. You go with 10 compounds into the clinical trials, maybe one comes out into the market after 7-8 years of the clinical trials. So that's costs about $2 to $3 billion. And that's very expensive and risky business, basically. And discovering a new drug for a unmet medical need or something. If you look at Alzheimer's disease, people have been many companies have been doing research for 30 years, but they still don't have a very good molecule yet. There are a couple of molecules that are approved, but I wouldn't say that they really are the block. Actually, can really help in Alzheimer's disease. All the patients, maybe in some patients, they're probably good. So this is where we end up in what is it? You cannot say that one size fits all. This is the bottom line. I was actually cruising through a uh, conference and called American Association of Pharmaceutical Scientists. There's this gentleman, a professor, well-known professor. He was also a consultant to us many times. I bumped into him, and he said, "Hey, Manny, we have a new model that we can explain how do we predict what happens in the humans." And I was curious enough, and we sat with it. There's another company called Hurel. That gentleman, the CEO of that company, also. They showed me a model where they have a microfluidic chip actually, and that they use and put different. It was a primitive model at the time, but they put different cell types in different compartment, like human blood is flowing in the humans beautifully. You can flow the any fluid into that and measure the various. You can measure the toxicity. You can measure the drug metabolism. You can measure protein binding. Lots of things that we do in vitro. You can actually do that in this microfluidic chip. So I got interested. We collaborated for 4 years on that. The data is really good. The first thing we saw is a flow system is very important. Until then, traditionally people were using a static 2D model cell culture where you take primary hepatocytes from the liver from humans or even animals, and you put the drug and look at the effects. But in a human, it's not just looking at human hepatocytes. This drug is circulating all over the body. It may be metabolized elsewhere. So there are also lots of things that communication between liver cells, especially parenchymal cells, and endogenous macrophages like Kupffer cells, endothelial cells, and they all have to be important. So using a single static cell may not be a viable option. That is where I changed my tone, and I said, "Hey, we need to look at human as holistically as a human, not in a single cell or a microsome which we isolate from the liver or something." But together, I'm not saying that we cannot just use them. What I'm saying is that you need all of them to be able to mechanistically elucidate the pathway of toxicity or even efficacy in very eloquently. So what happens is that in humans, as I said, we cannot do the human assessment in humans directly. You cannot dose a drug to humans and find out what happens. So these are the ways to you can actually understand what is happening to the drug in the body. What body drug to the drug is one thing, and what drug to the does to the body is another thing. So these are the two different pharmacokinetics and pharmacodynamics we call them. So basically, you have to shift the gear into investigative models. That's where 3D models come in. So I worked. I came back to India and I built infrastructures for two companies, Sai Life Sciences and Eurofins Advinas, to some extent. So, but I had this thing that on my back of my mind that I need to reinitiate these 3D models. So, fortunately, I had another friend who also was trying to start his own company, and then we collaborated. He was a friend of mine. He also was Dr. Uday Saxena. He was the chief scientific officer for Reddy Labs for about 10 to 12 years, and then he also wanted to change career. And then he when I talked to him about this, he was also very interested in getting into this 3D models. How do we build it? The question comes is that how do we do it? There's another technology, not only microfluidic chips, there's something else coming on board was 3D bioprinting. This is where I thought it's more important because microfluidics, it's very micro. Means it's a lot of challenges are there in microfluidics that analytically or even the mechanisms of toxicity to measure, a lot of challenges are there. So 3D bioprinting is more macro, and where you can actually put more cells into it, more, and you can measure a lot of different things, not just toxicity, all the other biomarkers, PD, PK, metabolism, everything you can measure in one shot in those 3D bioprinting. You can go up to 384 well plates as well. We have created 5 models so far. One is lung COVID model, where during COVID, people were looking for what other drugs we could use. There are lots of 7,000 or plus drugs are out there in the market. Can we salvage one of them to treat COVID? People have used ivermectin, hydroxychloroquine, but they were not approved by FDA. FDA said these are not the right molecules, so you need to look at something else. So that's where we looked at. Of course, we combined artificial intelligence there we cannot screen 7,000 drugs into this model. We shortlisted to 2025 them, and we got a very good data showing that this lung COVID model, the beauty of this lung COVID model, you can actually look at the pathophysiology of the COVID disease. That includes inflammation, cytokine storm, as well as thrombosis, which is why major problem is in humans is thrombosis, which doesn't happen in animal models. So whether you take rats, hamsters, or mouse, or any other species. So this is where we started the 3D models, and we were actually come up with 2 drugs in the process: a protein kinase inhibitor, ibrutinib, and a SGLT2 inhibitor, which is an anti-diabetic, is also very effective in preventing these 3 pathological events: inflammation, cytokine storm, and thrombosis in this COVID model. So we published this data in a BioRxiv, and we ventured on to other models like gut model. Today, microbiome is very important in terms of the diseases. People think that microbiome in the gut can go to liver, cause liver toxicities, and many other organs it may cause toxicities. So we created a gut model that's also published. We used an alcoholic liver disease as a model to study this, and we could show that there is a metabolite from the microbiome, which is a toxic metabolite, goes to the liver and actually enhances the liver toxicity induced by alcohol. So if you use antibodies against that toxin, you can actually reduce the toxicity quite a lot from the liver. So that's another one. We created a diabetes model where we could put in pancreatic cells, liver cells, and muscle cells together in a 3D bioprinting model and let them communicate with each other. So we actually got a very good idea on how to develop drugs for skeletal muscle disease. Okay, more than people are using pancreatic insulin secretagogues and etc. and other things. So today, majority of the Indian population is diabetic or pre-diabetic. About 400 million people, I think, are probably pre-diabetic, including pre-diabetes. So you do need more models and drugs. So that's another one. We also have a cancer in a dish model. Cancer in a dish model where we actually incubate the drug with intestinal cells, liver cells, and cancer cells together. So you have a 3 layers. So If you're taking a drug, it goes to the intestine first, gets absorbed, goes to the liver, and then from liver, if whatever is not metabolized goes to the target cancer tissue. So if I have to go to a patient, the doctor, and say, hey, doctor, there are 50 drugs for breast cancer. Which one would you start with? Right? He doesn't know. He'll try one first and then second, like that, they'll probably do. And in the meantime, patient is deteriorating, is getting more sick, more sick. So I said, this model we have is a great one that actually I can screen all the 50 drugs for breast cancer into this model and say which drug or drug combinations work for the best for this particular patient. So if you can give me a biopsy of the patient sample, I'll put it in my 3D bioprinting and tell you which drugs to use. So this is at the bedside, not at the bedside. It is not. We are not doing drug discovery research. We're talking about patients itself. We can actually tell patients with doctors, hey, use these drugs or drug combinations that will work better for this patient. So this is something we already have it, and we want to reach out to a lot of doctors and show them that this model can help uh, treat a lot of patients better. The other thing that we can also do is that a lot of genomics, proteomics, metabolomics, omics technologies are coming up and people are using it, but still people don't know which genes are responsible, which genes, there are lots of mutations that you can see. But in this model, we can also simultaneously look for the gene mutations and then stratify the patient populations and say, hey, this is what the drug is going to do for this patient versus that patient. So it becomes more personalized therapy for each patient. And this is where personalized and precision medicine, we can make it happen with 3D bioprinting or even microfluidic chips uh, in a nutshell. So this is what my journey has been. And we have other models coming up. And we can talk about them as we go along. Okay.
Thank you, Manny. It was so interesting, and I just got engorged in what the kind of science that you are speaking about. I mean, and you say that it is for bench. I mean, not for the research, but it's happening. It's translational stuff. So I think this is amazing. Well, as you were speaking and you were sharing your journey and about the kind of exclusive and extensive science that you have done, something was popping in my mind. That is, like, because you told about data analysis and putting in the 3D printer and stuff like that. What about artificial intelligence? That's a buzzword today. So is
Right.
there
Yeah.
so is there any
Right.
connection? Yeah, I mean, if you can tell us about it.
Right. So today, more than ever, that we want to avoid animals from research, whether scientific purposes or for ethical purposes. A lot of ethics are also involved in animal research. People say if only 10% drugs are getting into the market out of, I mean, 90% drugs fail, why are you still using animals? Fair question. I mean, there's no doubt about it. But at the same time, you're seeing that where are the models that other than the animal models that you can test it? This is where we are coming in and say, hey, we know the science behind it. We can actually create models in vitro, whether it's a static 2D model or a flow 3D model or a 3D bioprinted layer model. We can combine all these things and do an investigative mechanistic pharmacology and toxicology to bring more drugs uh, be successful in clinical trials. So this is one of the things. Then the question comes up is that what is artificial intelligence doing? I worked in artificial intelligence quite a long time. Usually, in the beginning, they were called in silico models. In silico means computerized models, right? I was exposed to this in 1993 while I was in American Cyanamide Company. It was very interesting. You know, you look at 1993, you're talking about 30 years passed already, right? So that requires a lot of data that you have to put into the computational models. There are different models. Some people use the chemical models, rule models, a lot of different things. I mean, uh, today is far better than 1993 in terms of the evolution of these models. But yet, I still think that there's not enough human data in these artificial intelligence models. These are the future, I'm telling you, because we don't have to do extensive animal research. We can only do very, very minimal animal research if we combine artificial intelligence with the 3D models. So here I am saying that our 3D models are human 3D models. We're actually doing human research. We're not doing animal research. We're not using animal 3D models anymore. We're using human 3D models. Take this data and put into the artificial intelligence. That you are enriching the human data, basically. So the predictive value of the artificial intelligence increases as you enrich the data that you need for the predictive value. So and the idea here is that the artificial intelligence is a buzzword, but how do you make it a better predictive model than it was in a lot of people are trying to think that they can just do computer model and can go into the clinical trial. No, FDA will not allow it. Okay, or any other regulatory agencies will not allow it because safety comes first. Without safety data, nobody will allow you to go directly administer a drug or compound into the humans. So you still need to do animal trials. This is where I'm saying that can revalidate that data in the human 3D models before going into the minimum animal trials. You don't need a lot of animal trials like you. I mean, if you start doing animal trials preclinically and of course non-clinically, sometimes you may have to do 1 year pharmacology studies, 1 year toxicology studies. At times, FDA may even ask 2 year repro tox studies if the drug is going to be given to a pregnant woman or if it is going to be has some hot spots on that it maybe can cause cancers in humans. So FDA will ask for more data. So you don't want to do 2 year CAR-T studies. Of course, today there are transgenic animal models that you could do a CAR-T study in 6 months uh, study rather than do a 2 year study. But yet, you're spending a lot of money, a lot of time going for 10 years to really release the drug into the market. And this is where the 3D models will probably accelerate the drug discovery research. And what I'm saying is 90% that drugs are failing, right? And I'm saying each drug to come into the market costs about $2 to $3 billion. Now, if I reduce that to 50% failures, I'm not saying I'll make it to 0% failures. It's not going to be possible. There still will be a lot of unknowns. So if I can reduce that to 50% failures, there you go. You can do at much cheaper cost, much faster rate, the drugs are going to come into the market. And that is where you can actually accelerate the drug discovery and development using these kind of drug discovery, artificial intelligence, and 3D, any in vitro model for that matter, that you can do it. Not only for drug discovery, I'm also saying these models can be applied to environmental toxicology. Today, we have a lot of pesticides, cosmetics, and many other things that people have to do animal trials. And of course, cosmetics in some countries, animal studies are banned, including India. So, but you still don't have the right models. There are skin models available, but there are some gaps in India versus Western countries. We have to import everything in India. This is what I'm campaigning for. Hey, I said hepatocytes I used 30 years ago. Artificial intelligence models I used 30 years ago. But the problem comes in India is that for me, the human hepatocytes, I cannot biobank them in India because there's no source I can biobank. Whereas in the US, companies are biobanking them hundreds of them, because there are policies, government policies are uh, not conducive enough. So they actually can, if anybody dies in an accident, uh, there are ways to get those organs. And there are some companies that are actually biobanking, non-profit organization, human brain cells, human liver cells, human kidney cells, pancreas cells. So if I want something that I want a human cell, then I can go to US to these biobanks and buy them. But I have to import them all at their cost, not my cost. So it becomes more expensive for me to do all that. So this is a bigger gap in India. I mean, if I know if I can get a human hepatocyte in India from some hospitals, human liver, I mean, I can isolate hepatocytes and I can cryopreserve them and I can even distribute them in India myself, so instead of getting them imported in India. So we also know how to characterize these human hepatocytes. Same way we can develop different models, different. So if we have biobanking, it's a very crucial thing India need to embark on it. Without which we can't do it, because if I have to get a liver and prepare hepatocytes, if I cannot cryopreserve them, then again I have to use them right away because you cannot freeze them unless you cryopreserve them. So these are the problems that we have, technical or some policy issues that we need to work on. Also, the instruments are very expensive in India. I mean, if you have to, if I have to use LC-MS for drug metabolism or flow cytometry for characterizing the some target receptor binding, etc., these are all have to be imported from the US. That kind of money for startups is not there uh, in India. So a startup, if they get 50 lakhs to 1 crore, that can vanish in 1 year to 1.5 years if you very carefully use them. But still, you cannot go to the, for example, BIRAC grants say that concept to uh, proof of concept, right? And you have an idea that you want to take it to proof of concept, they'll give 50 lakhs. But that's not going to be sufficient in the biotechnology industry. You really need minimum 2 to 5 crores to be able to do it. That kind of money is really not here. I mean, there are lots of 3D model companies in the US get $50 million to $100 million venture funding, venture capitals. We also don't have much of venture funding in India. So this is another thing that's another gap that we need to really, if lots of bright people in India, beautiful, brilliant people, but where they're stuck, not much of funding. So it goes to not just to me, everybody else who's starting biotechnology industry. Okay.
Right, right. So apt. I mean, that's a biggest challenge in
Right.
India when you start a biotech startup.
Yeah.
So when you were talking about all these things, so when it comes to clinical translation, from a clinician's perspective, I would ask this question. Uh, what about, like you talked about these in vitro, say, alternative models that we can use? Um, how do you think? I mean, can you give some insights regarding the accuracy and reliability of these models? And I mean, in any comparison that we can do,
Right.
because that's
So,
important.
right. A lot of people think these are predictive models. I say
Uh-huh.
no. I'm not using a crystal ball here to predict something that future. What I'm saying is that here I'm using a model that can give me insights into how to design appropriate clinical trials, more on translational clinical trials. We don't have that right now with the animal models. We are just going into the humans and see what happens with animal data. But here, these
Right.
models, 3D models, can help me get more insight, mechanistic insights. For example, we can generate a lot of biomarkers on the way for the disease related biomarkers or even safety related biomarkers. This is where omics come into the picture, from metabolomics, transcriptomics. I think transcriptomics is going big right now, more than the genomics and proteomics and metabolomics. So I would like to use those models in I mean, together with these 3D models, so I can get a more insights about what is this drug doing, not just doing efficacy, toxicity, but can also look at mechanistically what are the biomarkers I can use if I dose to the humans and get a better clinical translation. That's another thing that is coming. The second thing is that physiological based pharmacokinetic modeling, which I already mentioned in the beginning, where the human physiology is where very very important to do it. Our, our blood flows. I can't remember the rate right now. Per second, this much, per this many times. So your blood is flowing all the time through the heart, liver, brain, everywhere, right? That is what it is taking your food also, right? You're taking carbohydrates, proteins, amino acids. So all these things are traveling from one organ to another organ. So your gene expression patterns change each organ depending on which state it is. So it's basically what happens is with these models, we can actually look at the transcriptome changes better and understand what is happening to the drug when it goes into the body. Because here in my 3D model, I can take out the pancreas cells and see how much is actually dead, how much is actually still alive, and is the drug producing insulin through the pancreas or not. So these are the things easily can be monitored in vitro than in vivo. We don't have really good diagnostics yet. So these will become molecular diagnostics part of it. So the molecular diagnostics is another expanding field today. So we also call it companion diagnostics because it's not just doing diagnostic. Companion diagnostics actually to make sure that, or the other word is theranostics. Theranostics means a therapeutic value of the diagnostic, right? So these are all can be done with these 3D models, including artificial intelligence if we plug in. In the next 10 years, you'll see a lot of data that's going to come out, not just from our lab, probably from hundreds of labs all around the world.
Right. Yeah.
Yeah,
Yeah. So with all your perspectives, and you are doing such a wonderful work, so how do you look into the future with advancements, or how do you foresee this field where we are trying to integrate AI and human alternative models in terms of drug discovery? And what is your perspective? What do you think that how these can transform the industry as such?
right. So every model is a model at the end of the day, right? It's a
Hmm.
model.
Hmm.
You're trying
Right.
to build a model.
Hmm.
But the clinical translation is the most important thing. If you don't translate whatever with the model you use, like I said, animal models are not translating. We've been using animals for 100 years, but they are the only models we had to use. Now we are actually with modern drug discovery, right? I mean, as I said, today we have beautiful technology at our disposal that even 20 years ago we didn't have. People have sequenced the human genome. People have recombinant technologies. People have this 3D bioprinting. People have doing bioengineering, something I'm not into yet. But in bioengineering, you actually look at which matrix is very important for pancreas cells versus the liver cells versus. So lots of bioinks are being developed. The other thing I've mentioned that nanotechnology. Today, because many drugs that chemists synthesize, yeah, you have very beautiful data in the artificial intelligence model. But when you come into the reality, when you try to solubilize the drug, you take water and try to put the drug, it's not soluble. That's one of the major problems in drug discovery, that if you don't solubilize it, how is it going to go to the body? It may work very well if it goes into the circulation, but if it doesn't even absorb the circulation, how are you going to manage it? So nanotechnology is coming into the picture. I've already have one publication on how to use zinc oxide nanoparticles to as anti-cancer agents. Zinc oxides have been used in medical devices quite a lot. I'm saying that they can be used also. So nanoparticles, formulation nanoparticles, drug nanoparticles, and many other everything. If you can do a nanoparticles, you can also increase the solubility, and we can actually test in these 3D models and improve the bioavailability before you actually go into the clinical trial, so that you have better design of the clinical trial here, rather than just, okay, I'll just try to go and see what happens in the human. You go there and there's no drug in the circulation, right? You wasted a few $1 million into that. So I'm saying make informed decisions, not spontaneous decisions. That, okay, I want to go into the clinic fast. That is a bad approach. We need more informed decisions, do a very good clinical trial design, and accumulate the data in these 3D models. One of the things I want to talk to you about is if I have to do 1 month rat toxicology, or 1 month dog toxicology, or even 1 month uh, monkey toxicology, for example, it takes 2 years to finish this. I need kilograms of the material, drug material. Monkeys are 4 kilograms, and dogs are 10 kilograms. Of course, rats are only 200 grams. So I have to do 1 month for 3 doses up to 1,000 milligram per kilogram doses. You need enormous amount of drug, and that has to be prepared by the process scale up with no impurities, very minimum impurities and everything. That's a lot of work. Then you go and do 1 month toxicology. By the time you get all the data for each study, it'll take 6 months. Some people can say that they'll do it in 4 months, but realistically, it'll take 5 to 6 months. For rat, dog, and monkey, if I say rat, I do, and dog. So I'm talking about total, I need about 2 years to finish all that data. So what I'm saying is here, I need a gram material. I don't need kilogram materials. I can do all these trials in my in vitro systems in 6 months. And based on that, you can go to animal trials or even clinical trials. You can actually design a better trial with its informed data, rather than without any information out there. So this is where physiologically based pharmacokinetic modeling will also be applied, because we have a lot of physiological data here that we are incorporating it. Then we translate that better into the physiological system like human. And so we call these microphysiological systems. We don't call them uh, human physio, because we use in a micro level, as I said earlier, but a little macro also. I mean, but we still don't need a rat size or a human size to develop these chips. Okay, if I have to develop a chip of my size, forget it. We'll never make it. So what we're doing, so we need a chip that can actually enable translation better to the humans. Okay.
It was wonderful speaking to you, and we are almost at the concluding part of our conversation, Manny. So
Yeah.
this question I ask to all experienced people like you, that because many aspiring scientists are there in our country, as well as many good brains, as you said, who aspire to start their own startups, particularly biotechnology or pharma as such, life sciences as such. So what would be your message to aspiring startup
Right.
entrepreneurs?
So I again openly put out this message. Thanks for this opportunity you gave me here. That most pharma companies in India, they take Ready Labs tried it, but they folded it. Drug discovery operations. Lupin tried it. They folded the drug discovery operations. Piramal started it. They folded the drug discovery operations. The problem is that this is not going to happen in 1 day or 2 days. You need to put more money continuously for maybe 10 years, 15 years, maybe up to 20 years. So if I am at J&J, right, they'll never give up on drug discovery. As even with 90% failures they have, they still keep funding it. 10% of their revenues go into the R&D, innovation. So my message to either Dilip Sanghvi at Sun Pharma, or G.V. Prasad and Satish Reddy at Reddy Labs, or Arvindo or any other company. That what I'm saying is that start funding small companies, take that innovation, then you in-license that innovation and do the drug development. That is how that works. The other thing I also want to say is that academic industry collaborations are not prominent in India. At MIT and Harvard, all the Pfizer, Novartis, J&J's, Merck, everybody actually works with these academic institutes where they do target discovery. What I mean by target discovery is that you need a target. Unlike 30, 40 years ago, where we didn't have target models, now we have a target discovery model. We know which target to go after, which can have a better impact on disease uh, benefit. So in such cases, target discovery becomes so academia does more target discovery and industry does more make sure that some of those targets do become viable to take into the clinic and do the translation sciences. So industry and academia have to come together. That I'm saying. I'm also calling out to IISc, IITs, do work with industry rather than just doing silently in silos, doing your own work in your own labs. But India, if it has to grow, you need to break these barriers. In academia industry barrier, number one, pharma biotech barrier, startup barrier, see, big pharma startup barrier. We need to get rid of those barriers and we all have to come together and increase the drug discovery uh, success in India itself. We don't need to use American drugs anymore. If we do this, we have to discover our own drugs, and that is where we should be combining together all our resources from big pharma, innovative brains and startup biotechs, and academia. Brilliant brains too. There, they all came from brilliant backgrounds, and you need to combine all these brains together. I'm brainy, but if I work in my own corner separately, nobody knows what I did. So we need to come out together. And in the same process, we can also train young brains. A lot of people
Right.
are graduating with MS biotechnology, MS pharmaceutics, a lot of things, bioengineering. They all can actually chip in, use their brains to do a better job. Right? The younger brains think faster and better. Actually, they have better ideas. If I give mobile to my daughter, which I'm struggling how to open an app, she knows it in instant second. She'll open it. So we need to
True.
use their abilities to think better and do it. So we also need to get younger crowd into this venture. How do we change the India in terms of the big innovative form, especially in biotechnology, drug discovery, and pharma. We don't want to limit ourselves into the pharma manufacturing. Pharma manufacturing is okay that we can manufacture it and export it out there, but that doesn't put us an innovation uh, hub in the world. Okay.
Right, right. Thank you, Mani. Thank you so much. It was wonderful, and I am sure that, like me, my audience, everybody got very enlightened with what or all your insights, and I'm sure that eminent minds who are on the other side of the screen, they pick up your insights, and I'm sure that changes always happen, and that's the only
Yeah.
constant. And I'm
Yeah.
sure with
Yeah.
many people might get right direction, and I think it was wonderful. It is so wonderful to
Thank you,
speak to
Dr. Goswami. I really appreciate for the opportunity,
you.
including Geeta. Geeta actually spotted me at the conference, Indian Pharma Congress, and asked me to be part of this. Thank you, both of you, to bring me on board here.
Thank you, it is a pleasure. Thank you so much. Thank you,
Thank you very much.
everybody. Thank you,
Yeah,
and stay tuned
yeah.
for the next episode, and we will have some more insightful discussion. Thank you, Mani, once again.
Thank you. Bye.